The overall goal of this Duplex digital PCR assay is to simultaneously quantify general and human-associated fecal contamination in waters.
This assay can help answering key questions in recreational water quality monitoring, such as how much general fecal indicator, i.e., Enterococcus, and more importantly, human fecal-associated HF183 marker is in the waters. The main advantage of this assay is that it quantifies two targets in one reaction, and compared to qPCR, it eliminates the need for running standard curves and hence the associated bias and variability. Demonstrating the procedure will be Meredith Raith, our senior research technician.
To begin this procedure, first prepare 100 micromole per liter stock concentrations for all primers in molecular-grade water and probes in TEPH8 buffer as described in the protocol text. Next, prepare a master mix by mixing appropriate amounts of digital PCR mix, forward and reverse primers, the fluorescent probe, and nuclease-free water. Pipette up and down at least 10 times to mix while taking caution not to introduce air bubbles in the solution.
Since dPCR master mix is more viscous than conventional qPCR master mix, it's necessary to use the mixing by pipetting technique in order to create a homogeneous solution. This allows for accurate dPCR quantification downstream.
To make the assay mixture for droplet generation for running samples in duplicate, pipette 36 microliters of the master mix into a regular PCR plate. To each of the 36 microliters